131. The next one has plus plus, which is cis, R, D plus, which is parental trans, plus E is parental trans, and DE is recombinant cis. So this is a tetratype because there are 4 different types. The next one is plus E, which is parental trans, DE, which is recombinant cis, plus plus recombinant, and D plus is parental. So again, this is a tetratype. Then you have cis for all 4, which is all recombinant, non-parental ditype. And then the last one, you have D plus parental, DE recombinant, recombinant parental is a tetratype again. Okay? So for these, you see that parental ditype you have 145, and for non-parental ditype you have 135. So these numbers are, roughly equivalent to each other. The parental ditype isn't drastically larger than non-parental ditype. So this would indicate that D and E are unlinked. Now let's look at D and F. So I'm going to erase up here. And now we're going to look at D and F, so let's cross out E. Okay. In the parents, D and F are actually cis to each other, so parental would be cis, and recombinant is going to be trans. So the recombinant would either be D plus or plus F with respect to D and F. So the first one, all 4 have D and F cis to each other, which means it's all parental, and that is a parental ditype. The next one has them all cis, which is another parental ditype. The next one has plus F, DF, plus plus, and D plus, which would be recombinant, parental, parental, recombinant. So that is a tetratype. The next one has plus F, which is recombinant, DF parental, plus plus parental, and D plus recombinant, so another tetratype. The next one has D plus, plus F, and those are all recombinant, so that is a non-parental ditype. And finally, the last one has D and F cis on all of them, which again is another parental ditype. So if you add the total parental ditypes here, you have 145 plus 131 plus 3. You get 279. Now this is obviously way larger than 4, which is the number of non-parental ditypes. When you see that parental ditypes is much larger than non-parental ditypes, this indicates that D and F are linked. Okay? So now let's look at E and F. Again, I'm going to erase this one more time. And this time we're going to not look at D. Okay, so in the parents, E and F are trans to each other. So that means that recombinants would be cis, which would either be EF or plus plus. Okay, so the first one, they're all trans to each other, which is parental ditype. The next one, they're all cis to each other, which means they're all recombinant, and that is a non-parental ditype. The next one has them all trans to each other, which is parental. The next one has them all cis to each other, which is non-parental. The next one is all trans, another parental. And finally, the last one has 2 cis, 2 trans, which is tetratype 3. So how many parental ditypes? You have 145 plus 24 plus 4. So you get 173 parental ditypes. And 158 non-parental ditypes. So although there's some deviation, these are roughly equal enough for our comparison, and that would indicate that these are unlinked as well. And that makes sense. If D and F are unlinked and D and E are if D and F are linked and D and E are unlinked, that means that E and F must also be unlinked. So that makes sense. Okay. So now that we found E is on its separate chromosome and then D and F are on another chromosome, why don't we find the distance between D and F? Remember, when we're doing mapping in fungi, the distance between two genes is equal to the number of non-parental ditypes plus the number of tetratypes divided by 2 divided by the total number of asci times 100. Now remember that we're doing this with respect to D and F. So when looking at D and F, how many non-parental ditypes do we have? We have 4, right? Tetratypes, we have 24 plus 27 is 51. And how many total asci do we have? Again, remember, we have 334 asci. We'll times that by 100. So 51 divided by 2 is 25.5 plus 4 divided by 334 times 100. So we get 8.83 centimorgans between genes D and F. Now let's calculate the distance between each gene and its respective centromere. So when we're doing centromere mapping, which again remember we can do that because we're looking at Neurospora and they make ordered asci. So when we do this, what we have to do is look at each gene separately. So for now, let's write centromere mapping. And we're going to look at D, E, and F. So again, I'm going to erase what we did up top. And as Professor Schmidt showed you in the theory, when you're doing centromere mapping, you want to draw a line down the middle of each asci. And when you look above, if the 2 or 4 entities that are there are all the same, that means that it is an M1 first division segregation pattern, and that comes about from no crossing over happening between the centromere and whatever locus you're focusing on. If there are different, so say if there's 2 entities above the line you drew and they're different, that means you are seeing an M2 second division segregation pattern, which comes about from a crossing over event between the centromere and the locus that you're looking at. So let's look at D first. Here you see D, so they're the same above, and plus plus, they're the same below. So this is M1. The second one, you see 2 Ds above, M1. Now you're seeing a plus D above, which is different, so you're saying M2. M2 for the next. Now you're seeing DD, so that's M1. And the last one is also M1. So the important number that we want to know is how many M2s are we observing? Well, we're observing 24 plus 27, which is 51. So 51 M2s. And to calculate the distance between the centromere and this locus, you have to do percentage of M2s divided by 2. Well, how do you find percentage M2? There's 51 M2 out of the 334 asci, the total that we see. And to get the percentage, you're going to times by 100%. So you do 51 divided by 334, which is the total number, times 100. And you get 15.26. And now you have to divide that by 2, and you get 7.63. So 7.63 map units is the distance between locus D and its centromere. Now let's look at E. Okay, for E, the first one we see plus plus on top, so that's the same, and that's M1. The second one is also M1. Same for the third, because it's plus plus on top. Now it's EE on top, M1. EE on top is M1. And finally, the last one is plus E on top, which are different, so this is M2. So there's only 3 out of the 334 that are M2 for E. So we'll say 3 M2s. So what's the distance here? It would be 3 out of 334 times 100, and that whole thing divided by 2. So 3 out of 334 times 100 divided by 2. You see that E is 0.45 centimorgans away from its centromere. And remember that centimorgans is the same thing as map units. Okay, now let's look finally at F. For the first one, we see FF on top, so that is M1. Same for the second. Same for the third. Fourth. And now it's plus plus on top for F, so still M1. And then the last one's FF on top, so it's still M1. So if you see for F, there are no M2 second division segregation patterns seen at all. So 0 M2s, and that means that the percentage of M2 is going to be 0. So the whole unit is going to turn out to be 0. So there's 0 centimorgans between locus F and the centromere, which means that it's located within the centromere. Okay, so now that we know this, let's finally put together a genetic map as they wanted. So we know that gene E is located on a separate chromosome and is 0.45 centimorgans away from its centromere. So we'll put that here. On a different chromosome are genes D and F. So we just saw that F is within the centromere, and D is 7.63 map units apart from its centromere. So this makes sense. However, if you look above, what did we find here? We found that D and F are actually 8.83 centimorgans apart. Now in this case, usually, as Professor Schmidt has told you, that the lower number is more accurate, but in this case the higher number is actually more accurate because it includes single crossover events, but also some double crossover events that can happen between F and D. So for here, we're going to write that it's actually 8.83 centimorgans apart. Okay, so that's the genetic map for these 3 genes. Question 2. A yeast geneticist crossed a strain that requires adenine and histidine for growth, which is ad minus his minus, with another strain that does not, ad plus his plus. The geneticist opened 100 asci and grew colonies from the 4 spores from each. Cells from these colonies were tested for their ability to grow on plates containing minimal media, min plus adenine, min plus histidine, or min plus adenine and histidine. Analysis yield 2 separate groups. So group 1, from 78 of these asci, cells from half of the colonies were able to grow on all 4 media types, but the other half could only grow on the minimal media plus adenine plus histidine plate. From group 2, these were from 22 asci, and cells from one of the spore colonies were able to grow on all 4 media types. A second spore colony could only grow on minimal media plus histidine, and minimal media plus adenine and histidine. Cells from the third colony could only grow on minimal media plus adenine, and minimal media plus adenine and histidine, and cells from the remaining fourth colony could only grow on the minimal media plus adenine plus histidine plate. So it's asking, what are the genotypes of each spore in the two types of asci? So let's look at group 1. Remember that these were from 78 asci. It says cells from half of the colonies were able to grow on all 4 media types. Remember that one of the media types only contained minimal media, which means it didn't have adenine or histidine added to it, which means that the yeast had to have been able to make those themselves. Now to write this, that means that they were adenine plus, histidine plus. Now that means that they were prototrophic for both. Okay, and the other half of the colonies could only grow on the minimal media plus adenine plus histidine, which means that they were auxotrophic for both. So the other half were adenine minus and histidine minus. Okay, now if you just notice quickly, these are actually the parental genotypes. So these are all parental, which means this is a parental di-type asci. For group 2, this was out of 22 asci, and it says cells from one of the spore colonies were able to grow on all 4 media types, which again must mean that they were prototrophic for both adenine and histidine, so they were ad plus and hist plus. The second spore colony could only grow on the minimal media plus histidine and minimal media plus adenine plus histidine plates. So because only one of these plates has adenine and the other doesn't, that means that these spores must be adenine plus. But as you notice, they can only grow on media that has histidine, so they must be histidine minus. The third colony could only grow on minimal media plus adenine plus minimal media plus adenine plus histidine. So again, they only needed histidine on one of the plates and not on the other, so they must be histidine plus, but they needed adenine no matter what to grow, so they're adenine minus. And finally, the cells from the remaining colony could only grow on the minimal media with both adenine and histidine added, so they are ad minus and histidine minus. So what do these represent? Well, ad plus, hist plus, again, is parental, and ad minus, hist minus is parental, but the other two are recombinant. So if there are 2 recombinant and 2 parental in an asci, that means that these are the tetratypes. So these are the genotypes for each spore in 2 types of asci. For B, it says, are the adenine and histidine genes linked? If yes, determine the map distance between them. Well, as you can see, there were 78 parental ditypes and 22 tetratypes, but 0 non-parental ditypes. So 78 is obviously much greater than 0. And that indicates that these genes are in fact linked. Now, how do you calculate the map distance between them? Well, remember that the map distance is the number of non-parental ditypes, which in this case was 2, plus the number of tetratypes divided by 2, over the total number of asci times 100. So non-parental ditypes, there were 0. There were 22 tetratypes, so divided by 2 is 11. And in total, there was 100 asci. So when you times that by 100, you could cross both items out and do 22 divided by 2, which is 11. So there's 11 centimorgans between the adenine gene and the histidine gene. So they are linked, and there's 11 centimorgans between them. Question 3. A strain of mold that has blue compact colonies, wild type, is mated with a mutant strain that has green fuzzy colonies. The resulting tetrads were dissected to reveal the following. What were the parental genotypes? Well, we're saying that one strain has blue compact colonies, which is wild type, and the other has green fuzzy colonies, which is not wild type. So the genotypes, the blue compact colony, are going to be plus plus, and the green fuzzy colony are GR-FC. Okay, and now for B, it says determine if the genes are linked or independently assorting. Well, how do we do that? Let's look at the tetrads and see which ones are tetratypes, parental ditypes, and non-parental ditypes. Remember, for the parental genotypes are plus plus and GRFZ. So if you look over here to the third one, you have 2 GRFZ and 2 plus plus. Now these are all parental, so this is a parental ditype. Recombinants would be either GR plus or plus FZ. So GR plus, that's recombinant. GRFZ is parental. Plus FZ is recombinant. And plus plus is parental. So this has 2 recombinants and 2 parentals. So this is a tetratype. And finally, for recombinants, you have GR plus, that's recombinant. GR plus is recombinant. And plus FZ is recombinant. So these are all recombinants, so this is a non-parental ditype. So it's asking if the genes are linked or independently assorting. Well, we have 120 parental ditypes and only 4 non-parental ditypes. And because 120 is much greater than 4, we're going to say that these genes are actually linked. And remember, that's because if genes were unlinked, you would get a roughly equal amount of parental ditypes to non-parental ditypes. So these are linked. And then for C, it says determine any relevant map distances. Well, how do you find map distances in fungi? You have map unit equals the number of non-parental ditypes plus the number of tetratypes divided by 2 all over the total number of asci times 100. So how many non-parental ditypes do we have? We have 4. And then we have 12 tetratypes divided by 2 is 6. And total, we have 136 asci. So 10 divided by 136 times 100, you get 7.35 map units between these 2 genes. Between the gene for the color of the colony and also if the colony is going to be compact or fuzzy. So 7.35 centimorgans.
Question 4. 3 genes in yeast, di, mi, and tra, I guess we should say di, mi, and tr, are located on 3 different chromosomes. That's really important. A cross between a di/plus yeast and a plus/mi yeast yields an equal number of parental ditype and non-parental ditype tetrads, but no tetratype asci. Sorry about that. I wanted to underline that. Both di/plus crossed with plus/tr and mi plus crossed with plus tr yield many tetratype asci. Give an explanation for these tetrad results. Now, on the face of this problem, it could get very, very confusing. But as we're about to see, you are forced to go into a conclusion that may not be something you were thinking about immediately, but it's the only possible way you could get this type of data. So what I'd like you to ask yourself first off is, look, they're telling us, that's why I said it's very important. All these genes, each of them is on a different chromosome. And if you've watched the theory videos and you've solved some of these fungal mapping problems, we know that when genes are unlinked, right, as they're telling us these are, that the number of PDs should be roughly equal to the number of NPDs. And that's what we've always seen, and that's the way it is. And in fact, in some of these crosses, that's the way it is. But somehow, in at least a couple of these crosses, we are getting tetratypes. So we really want to ask ourselves is, when genes are absolutely indefinitely unlinked, how could you even possibly get a tetratype? Because when 2 genes are linked, how do you get a tetratype? You get a tetratype when crossovers occur in between the two genes, right? In fact, that's one of the ways we map. Remember, let's just make sure we have the concept down on this. If genes are linked, that doesn't apply to this problem, but it gives you a great context. You mostly see PDs. The amount of those is a lot greater than the amount of tetratypes, and the amount of tetratypes is a lot greater than the amount of NPDs. That's because in that context, NPDs are caused by a certain type of double crossover, right? But that's not what's happening here. So I'm just saying, normally this is the normal situation. If linked, you'll see tetratypes at a certain frequency. If unlinked, you shouldn't see any of them. Just for the record, so say we did this cross, this first one, Di plus and plus Mi, right? A parental ditype would look like this, right? Because there's two types in there, and they look just like the parents. So there's an ascus like that. An NPD, a non-parental ditype, would look something like, well, it would look like this, right? Because there are, again, two types, but in this case, they're not like the parents. So this is all background information that will really make it give us context to be able to solve this. And if a tetratype existed, let's just see what it would look like. There are a few ways it could be drawn, but it would be something like this. And if this seems easy to you, good, because that means you understand it. So we have 4 different combinations here, which is what a tetratype is, right? The order in yeast is not particularly important. So really what it's going to come down to is, how the heck do you get a tetratype when genes are unlinked? If it can't be crossing over between two genes, the only other thing we've ever discussed is crossing over occurring between a gene and its centromere. It might not be obvious why these are going to cause tetratypes in this situation, but let's look and see, because it's really the only thing that could happen. So let's look and see in the cross that we just did, all right, with the Di plus and the plus Mi, we know that there were no tetratypes made. So what that means is, let's just look with respect to Di, right? Remember, if you saw this, how we drew it, because remember, this is just Di plus. We're only looking at one set of, in other words, this is just the chromosome that has Di and plus on it. This is a heterozygote with respect to the Di locus, right? Normally speaking, let's just understand, if that's the only gene and there's no crossovers between the centromere, we're going to see things just as we would expect, like that, right? And if the same thing happened with Mi, if it was in the arrangement that we said, we're going to get either a parental ditype or a non-parental ditype, depending on how the segregation goes, right? So let me write over here, I'll write it the way we did here for a parental ditype. For the Mi locus, we'd have it like this, and that would give us a PD, right? Because the same type of thing that we just drew over here is going on with that other chromosome. But let's ask ourselves a question. What if a crossover did occur between that Di locus and the centromere? How would that affect? It wouldn't have any effect on this over here. How would it affect the arrangement of things over here? Well, it doesn't affect this sister chromatid, so that's going to stay the same. But what's now attached to this centromere is a plus. So I'll just draw it right next to it. We'd have a plus there. What's attached to this centromere is a Di, right? And a plus. So this is the arrangement that we'd get if there was a single crossover between the Di locus and the centromere, its centromere, which has nothing to do with these guys, but they're staying the same. So now if we put this all together into one ascus, voila, there it is, a tetratype, right? So even though the genes are unlinked, this is really the only way to get a tetratype under these circumstances. Now that we have an understanding of that general principle, let's look at the data. All they say is give an explanation for these tetrad results. So obviously, some kind of crossover is happening between a gene and in its respective centromere whenever we see tetratypes, right? So there's 3 crosses here. Let's carve out a little spot, and I'll change the color. They told us, here's cross number 1, di plus times plus mi. And if you do that, PDs equals NPDs. That's just what you'd expect, right? That means there's no crossovers between any centromeres. There's no crossovers, period. Let's look what that means. So here's the A chromosome where the DI locus is on there. I'll just draw one of them, right? Because pretend you were making a map. If you wanted to map that in the centromere, what does it really mean? In other words, if you're doing these crosses and you don't see any tetratypes, it means that very rarely or never does a crossover occur between this DI and its centromere. So even though I've drawn it over here, what it means is DI either is the centromere, which is questionable whether that's possible or not, or it's right on top of it, so there's no room for crossovers to occur in between there. The same goes for MI, right? Now don't get confused. This is a different chromosome. It's a whole different one. But MI is on there, and it's very closely linked to its respective centromere, right? So you'd say that both the loci DI and MI are tightly centromere linked. They're linked to different centromeres. Why? How do we know that? They're linked to different centromeres because they told us that they're on 2 different chromosomes. They're on their own chromosome. So without going through all the technicalities of this, if you cross this same DI plus times plus TR, or if you cross the MI plus times plus TR, they're saying that you do get a lot of tetratypes. The only explanation for that is that in fact the locus TR is very, very far away from its centromere. Remember, it's a different chromosome again, chromosome number 1, 2, and 3. But during meiosis, the way we've drawn it over here, there's plenty of room for crossovers to occur in this interval between the TR locus and the centromere, leading to a situation just like this and causing, in other words, any other chromosome, any other tightly centromere linked gene is going to give you tetratypes if you cross it with plus TR, or cross it with something involving the TR locus where you're able to have this type of tetratype situation. So I think the problem with this type of a problem is making automatic assumptions. And I do it too. You just have to be careful. When I see tetratype right off the bat, I think, okay, well, tetratypes have to do with map distance between 2 genes. But then they told us these are unlinked. So where did the tetratypes come from? And that's the only possible explanation. You could probably go into a more detailed analysis of this, but we answered the question. We gave an explanation. And even though I did the example here with the DI locus, where that's really happening is in fact with the TR locus, right? So if we want to just go down a little bit and write this out, we can say TR is far from its centromere DI is very close to its centromere. It's a different one, right? And MI is very close to its own centromere. That's the explanation, guys. Hopefully that makes some sense. Give it a little time to sink in, think about it. I'll leave the answers there, and I'll see you soon.
Question 5. The gene GU3 in Neurospora encodes for an enzyme involved in the synthesis of guanine. A wild-type strain, GU3+, was crossed with a guanine-requiring strain, GU3-. The resulting diploid went through meiosis and 100 asci were dissected. determine the distance between the GU3 locus and its centromere. So when you're doing centromere mapping, and remember that you can only do that in Neurospora because they make ordered tetrads, what you want to do is draw a line down the middle and look above the line. If all four above the line are the same, or GU3 plus or GU3 minus, that means that it's an M1 first division segregation pattern, which comes about from no crossing over between the centromere and the locus. However, if half of them are, say, plus and the other half are minus, that means that it's an M2 division pattern, which means that a crossing over event did happen between the locus and its centromere. So let's look at the first one. All of them are GU3 positive, so this is M1. The next one, they're all GU3 minus, which is M1. Remember, it doesn't matter what is above the line as long as all four are the same. So the next one, half are plus and half are minus, which means it's an M2 segregation pattern. Same for the next one. Then the next one is half minus, half plus, M2. And then half plus, half minus, another M2. So how do you find the distance between the locus and its centromere? You take the percent of M2 segregation patterns divided by 2. Now to find percent M2, that is the number of M2, which here is 14, divide by the total number of asci, which here is 100, and then times it by 100% to get the percentage. So because you're dividing by 100 and multiplying, you can just cross that out and get 14. 14 is the percent of M2 in the total asci. And when you divide that by 2, you get 7. So there's 7 centimorgans between the GU3 locus and its centromere. So if you want to draw a chromosomal map of this, you could draw the centromere as a circle. Say this is the GU3 locus, and there are 7 centimorgans in between. Problem 6. When the following fungi diploids are sporulated, indicate the percentage of tetrads that would have 0, 1, 2, 3, or 4 viable spores. For A, the fungi diploid is a pure breeding wild-type strain. So for this, if it's pure breeding and wild-type, you can write its diploid genotype as plus, plus. And when this strain goes through meiosis, all tetrads, so 100% of the tetrads, will have 4 viable spores. And that's because each will get a wild-type allele. For B, it says a strain hybrid for a null mutation in a single essential gene. So it's hybrid, so that means that it has one wild-type allele and one null allele, where N is for the null mutation. So the diploid is viable because it has one correct copy of this allele. However, when it goes through meiosis, 100% of the tetrads, or all of the tetrads, will have 2 plus gametes and 2 N, because remember they separate, and these 2 N's will not be viable because they don't have this essential gene, which means they can't make this essential enzyme, whatever it is. So 100% of the tetrads will only have 2 viable spores.
Let's take a look at question 7. In yeast, CL and FN are heat-sensitive mutations of different essential genes. For each part of this problem, a CL FN plus times CL plus FN cross was done under low temperature conditions so that all yeast would be viable. Under high temperature, lethal conditions, indicate the percentage of tetrads that would have 0, 1, 2, 3, or 4 viable spores. Okay guys, so basically what they're telling us here, it's very important obviously to get down what do we know and what are we being asked. These genes, whatever they are, CL and FN, they are essential. That means you need a working copy of both of them for cells to be alive. So the CL or FN mutation, if either one of them is mutant, you're going to not have a living cell. Since they're heat sensitive, this will only go into effect when the temperature is raised to the so-called lethal or restrictive temperature. Okay, so I should say that it's very important to understand, if you're not doing these in order, question 4 in this section has to do with how you get tetratypes when genes are unlinked. You have to understand that in order to get this, because we can't go through it all again. So take a look at that if you haven't already. Let's check out A. So they're giving us different scenarios and telling us, in this case, if this was what the situation was, tell me how many are going to be, well, exactly what they asked. What percentage of the tetrads that would have 0, 1, 2, 3, or 4 viable spores? What does viable mean? Let's just be clear. Only viable is like this at the lethal temperature. Both genes plus, both genes working, right? Okay, so if the mutation loci are unlinked and they're both located within their respective centromere, this means, if they're unlinked, right, it means that PD equals NPD. And the fact whether it's true, really reasonable or not, that they're in their respective centromere, what that's telling us is that no crossovers are occurring and that no tetratypes are going to be around. So we're going to have half PDs and half NPDs. Given the cross, PDs are going to be like this, right? Because we know PD is a parental ditype, right? There's 2 types in there, and none of them are viable. 50%, half of the tetrads are going to be that way. Now, an NPD is opposite in the sense that we're going to have only 2 types, the double mutant and the double wild type. They're 2 types, but they're not like the parents, right? And the ones that are viable here, remember we said this is what we're looking for, are 2 spores down the bottom here. So really, I mean, it's not that bad. For part A, 50% or a half are going to have 0, the PDs, 0 viable spores, right? The other half, the NPDs, are going to have 2 viable spores. So let's go on to B. If you want to look at any of these a little longer, just hit pause, because I'm going to erase it rather than go down so we can keep the questions here in our site. Okay. 50% 0, 50% 2 for that one. How about B? The mutation loci are unlinked. Okay, CL is located within its centromere, but FN is 20 centimorgans or map units from its centromere. So again, we can't go into all the details about this, but if FN is 20 centimorgans from its centromere, that means a certain amount of crossovers are going to occur in that interval as the cross is set up. Remember though, what map distance is in this sense. In other words, the formula that you learned was that the map distance is the percentage of, well, the way we did it with the centromere was the percentage of M2, but basically that referred to crossovers. So the percentage divided by 2. The bottom line being, if it's 20 centimorgans as the distance, you now have to multiply that by 2. It means 40% of the resulting tetrads will be tetratypes. Now, that's really important to get that, obviously. I didn't go into a full explanation, but the bottom line is that you need to know 2 things here, which we can't go over 100% in this problem. One is that the only way to get tetratypes when things are unlinked is if there is a crossover between one of the loci and its centromere. The other thing is just knowing the fact that map distance equals the percentage of a certain type of tetrads made by crossovers divided by 2. So we have to multiply by 2 to get the 40%. So this is going to be a tetratype. The 60% that are left, they were unaffected, and since things are unlinked, those are going to be PDs and NPDs, right? We just said a minute ago, so there's really no need to draw it all over again. In a PD, there are 0 spores that are viable, and in an NPD, there are 2. Since half and half, we're going to have 30% of these and 30% of these. But let's draw out a tetratype. So it's going to have one like the parent on top, one like the other parent on the bottom, then a double mutant, and a double wild type, right? And only that one spore is going to be viable under the restrictive conditions. So what we could say is 40% of the tetrads are going to have 1, that's the tetratypes, right? 1 viable spore. 30% are going to have 2, that's the NPDs, and 30% are going to have 0. Those are the PDs. That's a little harder, I suspect. Okay, so again, pause if you want to keep looking at that, because I'm going to erase it. This is fun stuff. It is, actually. If you think it's fun, don't worry, you don't have to admit it, but you could admit it to yourself. C, the loci are 0 centimorgans apart on the same chromosome. Again, this may be very, very unlikely, but in essence, if they're 0 centimorgans apart, in essence, they're the same gene, right? I mean, what that really means is that in a cross between the two, it's the same gene. Everything is going to be a PD, no? I mean, if we're under the assumption that they are, in fact, the same gene, it does run into certain problems. So you have to say, look, they're different genes, but no crossover could ever happen in between them, no matter what. And technically, they are linked, right? So when they're linked, the only way to get an NPD is to have a double crossover. So you're not going to get any crossovers at all. So no tetratypes, no NPDs, all PDs. And we already just said before that PDs don't have any viable. So here we have 100% of the tetrads having 0 viable spores. I don't want to say that's easy, but once you understand what they're really saying, it's a little dumb, I gotta say. But when I say dumb, I just mean, you know, I'm sorry, I'm sort of dumbfounded just trying to explain it. We're telling you there's 2 different genes, and they're saying that they're right on top of each other. It's not impossible, but it's very, very unlikely. But they want you to understand, so they can ask. Here, what about the last one here? D. The loci are 20 centimorgans apart on the same chromosome. Assume there are no double crossovers. So that means we'd have a situation wherever the centromere is irrelevant, in this case, where CL and FN are 20 map units apart. Now in this case, you still have to multiply by 2 because basically when there's no double crossovers, it's the number of tetratypes over the total, which is the percentage divided by 2, sort of just like before. So that means when there are one crossover, when there are no crossovers, you get PDs. When there's one crossover, you get tetratypes. And if there are 20 centimorgans apart, just like before, 40% would be tetratypes. The remaining 60% would be parental ditypes. And I don't think we have to draw it out again. PDs have 0 viable spores. Tetratypes have 1. So 60% have 0, 40% have 1. I mean, these are either mind-blowing or not so bad, depending on your ability to sort of put everything together that we've been doing. Certainly, you couldn't do this. I'm not saying you couldn't, but it would be very difficult to do this problem if you didn't already have a lot of background in your mind about how to do map distances in fungi, etc. All right, good job on that one.